earlier session, we discussed the various types of cleavages that the zygote has to undergo so as to give rise to further stages of embryo. When we talk about the types of cleavages, the ones that we discussed earlier, they were based on what they lead to or how is the direction of the cleavage. There are a few more ways involving the kind of symmetry they lead to. Like for example, bilateral cleavage. See, as we have seen, this is the zygotic cell which divides meridionally to result into two blastomeres. Now, each one of them would divide further. And if it divides in such a way that every one gives rise to structure that is symmetrical on both the sides. So two symmetrical paths are being developed, then we call that as bilateral cleavage. Suppose the other type that we will discuss is radial cleavage, which would mean say the same thing. These two daughter cells have been formed. Each one divides so there are two daughter cells in the lower half, two daughter cells in the upper half. If that continues in further stages, then we call that as radial cleavage. Whatever number of cells you find in the lower half, same number you will find in the upper half. Bilateral cleavage would be found in tunicates. Radial cleavage would be found in Amphioxus or even amphibians. But in amphibians, let's understand that in later stages, megameres or micromeres will be distinguishable. Then there is something called as rotational cleavage. That would mean, suppose if this is what you are seeing in top view, these are the four daughter cells that you are seeing. In the next cleavage, this is how four more daughter cells will be formed. Okay, they are not exactly below these. That's called as rotational cleavage. Which is found in most of the mammals. Whereas there is something called as spiral cleavage. That is, if these are the two daughter cells, the next daughter cells are found in this manner. Next daughter cells will be found here. So they are spirally arranged. That's called as spiral cleavage, which is found in annelids or even flatworms. Now that's on the basis of the symmetry they result into. Here, if you talk about the blastomeres, up to a particular stage, the fate of the cell, the future of the cell is yet to be defined. We call these as indeterminate cleavages. The fate is not defined yet. Whereas if these cells have a defined future, defined fate, then you call that as determinate cleavage. That's how cleavages can be defined on the basis of symmetry and fate of the cells. Now let's know what exactly is this going to lead into. In general, every multicellular animal, it starts developing, say, from a zygote, which divides first of all into two daughter cells. These two daughter cells will divide further, give rise to four daughter cells, and this will continue till it leads to formation of 16 cells. Now, usually up to 16 cells, the cells have not defined fate. No defined fate is assigned to individual cells. That means undifferentiated cells would be there in 16 cell stage. That is called as a morula stage. Morula stage. Whereas after this, the cells have a distinct future to give rise to. What that cell will give rise to, which germ layer, which tissue, which future organ, that is defined after that. So after this, it would lead a stage that's called as Blastula. Now let's know why it's called as morula. Morum, it's a Latin word that stands for mulberry. So here it has a mulberry-like appearance. All cells almost appear to be similar. That's therefore called as morula. While here is called as blastula because there's a distinct cavity surrounded by one single layer of cells. These cells are known as blastodermal cells. Derm stands for skin. Blast stands for fission, division. So a layer formed by division of cells, hence blastoderm. And the cavity they enclose is called as a blastocele. Cele stands for the cavity. So blastocele surrounded by one single layer of blastoderms, hence that's called as a blastula. Up to blastula stage, all the animals go through similar process of development. As I said, the types of cleavages can vary. The types of egg cells will be different. That is mesolecythal, microlecythal, whatever. Accordingly, there's going to be a difference in the developmental process. But broadly, this is what the embryos will be going through. Now this blastula, there can be different types of blastula. And these types are made on the basis of the cavity and the cells around them. Broadly, what it would look like, this is a layer of cells and this is the cavity inside. Okay, the layer would be, as I said, blastoderm, and this is the cavity is blastocele. This would happen predominantly from the microlecythal or alecythal cells or alecythal egg cell. If it's meso or megalecythal cells, megalecythal egg cell, and if it undergoes holoblastic unequal cleavage, then it may result into this kind of structure. Eccentrically positioned blastocele. Upper three sides are covered by micromeres, the lower side is covered by megameres. Okay. In both the cases, you are seeing the structure to be consisting of a large cavity. A large cavity. Prominent part of the sphere is occupied by a cavity. So this is called as coeloblastula. Cele stands for the cavity. So coeloblastula are a blastula with a prominent blastocele. This is found in most of the amphibians, as we have seen. This is case of frog. sometimes we find that the cavity is much smaller, hardly negligible in size. Most of the sphere is occupied by the cells, the blastomeres. Then in that case, that blastula is called as Stereoblastula. Stereoblastula. Example would be molluscans or nereis. That's an annelid. Then in the cleavages, we have seen a type of cleavage as discoidal cleavage. That is, only large amount of yolk is contained, and the animal pole, or let us say cytoplasm and nucleus, are only on one side, and it undergoes cleavage only at that part. A disc-like region is found there. That's called as the discoid cleavage. It results into a blastula, which is then called as discoblastula. That would mean, suppose if this is the sphere that we are referring to, this is the cavity, these are the cells, blastomeres. A disc-like structure is being developed, so that's called as discoblastula. Example would be bony fish, reptiles, or birds. Birds, we would be able to relate it a little later. Then the other type would be superficial blastula or periblastula. This is also found in discoid cleavage. But here, suppose if this is the sphere that we are referring to as blastula, the cells or whatever cavity, it's related to only this part. The cavity and blastomeres, that will be only related to this region. And that's called as superficial blastula or periblastula. One unique type of periblastula would be seen in the case of insects, wherein it's a meroblastic cleavage, let's say. This is the cavity, and the cells are being developed only in the periphery. Rest all is the cavity. That's called as periblastula. There's something called as amphiblastula. While in mammals, there is a stage called Blastocyst. Now, what's different here? Here, the peripheral layer of cells these cells are called as trophoblast cells, or that layer is called as trophoblast layer. And inside this would be a mass of cells. These are called as inner cells, or these are actually embryonic cells. And the cavity that we're talking about is this cavity, but now we would name it as a blastocyst cavity. Blastocyst cavity. So trophoblast would be the outer covering. Inner mass of cells, central cells also you can call it. would be the embryonic cells, and in between there's a cavity called blastocyst cavity filled in with a blastocyst jelly. That's found in mammals. Human being could be a typical example. Now that's classification of stage called blastula. And earlier we saw the different types of cleavages. The stage after blastula, wherein the fate is more predominantly developed, almost the germ layers have started appearing, and it's going to result into organogenesis or morphogenesis. That stage is called as gastrula. Now, formation of gastrula from blastula we would talk about in the next session. I'm sure you must have liked this video. Share it amongst your friends and family, subscribe to it, and don't forget to post your queries, suggestions or comments. Thank you.